So much, Kira, for the kind introduction, and thanks so much to the organizers for inviting me here. It's really amazing to have a real life conference, and I'm really enjoying it, even though it's a bit different than in other times. So what I want to do with you today is give an outline of some of the developments that we've been working on in my group together with collaborators on computational genomic approaches to study biosynthetic pathways, and specifically those approaches that are relevant for the study of diversity, genomic diversity of megasyntheses, PKSPOP syntheses, and non-ribosomal peptide syntheses. So my group really focuses on methods to identify biosynthetic gene clusters that encode the production of natural products, and to use large-scale omics data to chart natural product biosynthetic diversity, and to also link this to chemistry, because of course the genes, the genetics, and the chemistry are functionally tightly linked, as you all very well know. And to this end, over the past years, we've, together with collaborators, developed a kind of comprehensive workflow to analyze large-scale genomic data for natural product biosynthetic diversity. And it basically starts with where also my scientific career started, with the ANTISMASH pipeline, which identifies biosynthetic gene clusters. Then we have MIBIG, which is an online data repository for known biosynthetic gene clusters with known products that have been experimentally characterized. And then, since recently published earlier this year, we have tools, for example, BigScape and COSOM, that allow really exploring the diversity of these gene clusters at large scale. So what BigScape does, it takes gene clusters identified through ANTISMASH together with reference clusters from MIBIG, and then it builds sequence similarity networks to really show and visualize how all of these gene clusters are mutually related to each other. So gene clusters, pairs of gene clusters that have similar domain architectures, similar sequences, they will be linked in the network, so that the nodes will be gene clusters, and the edges that connect them indicate a high level of similarity between those gene clusters. And well, if you actually use this on, let's say, a couple of hundreds or thousands of genomes, this really allows you to get like a bird's eye overview of the diversity that is beneath that. And then what a tool like COSOM does, and this was made by a PhD student from Paco Barona Gomez's group in Mexico. What COSOM does is that can take, for example, a gene cluster family, and then make a multi-locus phylogeny of all the underlying gene clusters to also show how they are evolutionarily related. This is also built into the BigScape workflow as well, but you can also use it completely standalone. So let me illustrate how this works, how an output of this looks like, what it looks like. This, at the start, you'll see like a visual overview of all the genomes for all the gene cluster families that have been detected by BigScape. This visualization was created by Safia Gozar, a PhD student in my group. And then, for example, you can go to the type 1 POLYKETIDES, and you'll see all the gene clusters of this type represented in a visual output. And then you can actually hover over with your mouse, or you can use the search box to, for example, find certain reference clusters based on known molecules that are represented in the MIBIG database. And for example, you can then find all gene clusters that are related to a known cluster of interest, and also see their evolutionary phylogenetic relationships. You can also search in various other ways. For example, you can search by taxonomy. So here we go to GRIPs instead of the type 1 PKSs, and you can search those in several ways. So you can search by taxonomy, for example, genus name, and they will light up in the network. Or you can search by a certain protein domain, and for example, an enzymatic domain of interest. And also then they will light up in the details. You will then see all the gene clusters that have that protein domain as a part of their biosynthetic gene cluster. And this, if you have a very large data set, for example, a strain collection, and you sequence all of their genomes, this allows you to really rapidly explore the gene cluster content of such a strain collection, for example, and identify gene clusters of interest and their relationships to other gene clusters. So when we published this work, we collaborated with the groups of Neil Callagher and Regan Thompson to show that you can actually apply this also to actually find novel biosynthetic diversity. We applied it on a group of biosynthetic pathways, which are also megasynthases, NRPS, PKS systems that are involved in making detoxins and bromosamides. So these were studied for a few years already by the groups of Neil and Regan. And so they had already identified two clades. So one is the toxin clade and one is the bromosamide clade, for which they had experimentally characterized representative gene clusters and identified the structures of their products. So from our analysis using big scale and Colosson, it became clear that there were a number of additional gene cluster families, clades of these types of gene clusters that were clearly related to these two known ones, but they were slightly different by having different tailoring enzymes or also differences in the sequences of the NRPS and PPS modules. So Michael, he was a postdoc with Neil and Regan, actually did experimental characterization of a number of representatives of these. And here you see a Coruscant tree of all these gene clusters. And so the known clades before that time were here, the bromosamide clade and the toxin clade here, and representatives of, representatives of these three other clades. So the one here at the top, supercluster clade, and the Amyloidopsis clade, were experimentally characterized by Michael. And for example, here we saw that there's an additional cytochrome P450 encoded next to the core of the gene cluster, as well as an enzyme reductase. And this the toxin happened to have like an additional heptanomide chain. In this clade, which is a clade of clusters that go together in kind of a supercluster configuration with amino glycoside gene clusters that are right next to it. Michael saw that actually different substrates are incorporated by the NRPS part of this pathway. So we get, instead of phenylalanine, we get tyrosine or phenylalanine. And in this clade here, from Amyloidopsis, there is an additional cytochrome P450 hydroxylase, which actually hydroxylates the valine in these toxin molecules. So this really shows that if you go systematically chart the different subfamilies of this bigger group of enteroprotic biosynthetic gene clusters, you can find all sorts of interesting variations based on, for example, different tailoring enzymes, differences in structure specificity. And I think it's really interesting to think about this also in terms of, for example, enteroproduct optimization. Because when you systematically identify all the biosynthetic variants of an enteroproduct biosynthetic family based on this, it would then also be possible to start combinatorializing these by, for example, taking combinations of tailoring enzymes from the different clades to make even additional libraries of variants of such molecules. I think this, I'm very glad that this really constitutes powerful technology to really use these thousands and tens of thousands of genomes that are out there to really make discoveries. But when we finished this, we did realize that we did want to make it even bigger than BigScape, in the sense that we wanted to also be able to do this at a global scale. So BigScape through a number of algorithmic innovations quite fast, and you can really use it to easily analyze a couple of hundreds or thousands or most tens of thousands of genomes. But when you go like above like 50 or 100,000 gene clusters, you get into the computational limitations because of the sequence similarity networking, which involves a lot of pairwise similarity computations. So we thought it would be really cool if we're also able to do this and identify gene cluster families in a global fashion. For example, taking all the genomes that are out there, which are now hundreds of thousands of genomes, more than 1 million gene clusters, and cluster those into gene cluster families. So this was a project led by Satya, who was also involved in the big scale visualization. And Satya, what he started with is he tried to find a way to kind of represent sequence diversity without actually looking at computing sequence similarities or sequence identity itself. And he did that by using protein domains like we have done before, but then in a more accurate and more high resolution way by coming up with sub-PFAM domains. So if we would just use general PFAM domains, then for example, an NRPS annihilation domain would be the same across all the NRPSs, and it wouldn't give you very much information to know that A domain is part of a gene cluster. So we need to know it is an A domain, but is it a certain subtype of an A domain? So what he did is he first identified a range of relevant protein domains that occur inside biosynthetic gene clusters, both from PFAM and from the library of domains that we use in ANTISMASH. And for more than 100 protein domains and key enzymatic protein domains that occur frequently in various classes of biosynthetic gene clusters, he did a large-scale phylogenetic analysis to identify subfamilies of these protein domain families. And for each of these subfamilies, which you color here around the tree, he built a sub-PFAM model that identifies the subclade that is represented by it. So in this way, we can identify, without doing sequence-to-sequence comparison, we can identify specific clades of protein domains in a gene cluster in a very fast way. So what Satya then did is, well, he applied this to a very large data set of 1.2 million gene clusters. So then you get a big matrix, a feature matrix of gene clusters here versus domains and subdomains here. And then he used a clustering algorithm called the Birch clustering algorithm to be able to identify gene cluster families. And because this subdomain presence and absence, and also the kind of whether it's the best hit or the next best hit to the subdomain models, because these are discrete values, you actually this way featureize you vectorize your gene clusters in such a way that you can, for each gene cluster family, you can identify basically a point in gene cluster space where you say, okay, this is the central point, and all the gene clusters around this point we call to this gene cluster family. And in the end, this really is scalable to millions of gene clusters. And if we look at the computational benchmarking of this, Satya also compared this with the big scale. If you go to very large data sets, hundreds of thousands of gene clusters, you'll see that big scale starts to get into trouble with very long runtimes. If you go indeed above like 100,000 gene clusters, well, big slice runtime stays very low and scales much better. If you compare the distances computed by big scale and big slice, the correlation between them is pretty good. So what this also allows is doing global analyses of biosynthetic diversity across all genomes that are publicly available. And we're currently working on this, or helping Nadine's group in tackling this and doing this in a systematic way. And we're really excited that this way it should really be possible to identify really, for example, which taxonomic groups have the largest capacity of making different kinds of natural products. There is one caveat when applying this to NRPSs and PKSs, is that sometimes big slice does tend to lump some of these megasynthase gene clusters together in big gene cluster families if they have a lot of core similarity. But we do see that underneath, if you basically look into patterns within such a gene cluster family, so here there is a number of curated groups that we know by manual annotation they should belong together. We do see that, for example, within this gene cluster family, the ones with the same color, they do tend to go together, so the underlying patterns do make sense. So this means that you can actually deal with this by either using a stricter cutoff to break up this gene cluster family in multiple subfamilies, or, for example, by running BigScape on the big slice families to get a high resolution analysis on the inner relationships of such a still pretty broad gene cluster family that you would get with a big slice. So, since a few weeks, we also have a database available called the Big Fan database, where you can actually access the gene cluster families computed by big slice. So, indeed, we took all the gene clusters from both genomes and also metagenome assembled genomes, and also represented by and linked to the ANTISMASH database. Then, so that's in total 1.2 million gene clusters. These were clustered into gene cluster families by big slice, and the gene cluster families are stored in the Big Fan database. And what makes this really usable as well is that you can, for example, if you have your favorite gene clusters that you detect using ANTISMASH or FungiSmash, you can actually query your gene cluster to the Big Fan database, and using the same computation, the same vectorization, Big Fan will assign your gene cluster to the nearest gene cluster family. So it will show you which gene cluster families in the database are most closely related to your gene cluster of interest. So this way you can quickly get an overview if your gene cluster is part of a larger family of gene clusters, which perhaps known gene clusters are also part of that gene cluster family, etc. And really make use of the full width of all the genomic information that's out there. So when you're able to do this, it's still very nice that you're able to start with these very large data sets, but in the end, of course, you will all want to zoom into specific gene cluster or specific biosynthetic pathway of interest, because that's where indeed in the end the magic happens and new chemistry is found. So you'll probably find your gene cluster, analyze it with ANTISMASH or another tool, and try to see whether this is an interesting pathway to experimentally characterize, whether there are some predictions that you can make about the chemical structures, etc. So I want to highlight in the second part of my presentation a few developments that are new and upcoming with regards to predicting, for example, chemistry, and also in the context of PKSs and NRPS systems. So Eric yesterday already introduced you to the TransPact algorithm, which annotates trans-AT PKSs by assigning ketosynthase domains to functional plates and being able to make dendrograms of how the polyketide synthase assembly lines are related to each other. So this is also scheduled to be integrated into the next version of ANTISMASH. Perhaps also with the TransAIDER tool that Eric also developed. That's still something we need to figure out a bit. We're also working on a new algorithm for NRPS subset specificity prediction. Barbara and my group is working on this. I think this is the way we're doing this is going to be really different from the way it has been done before, but I'll not have time to go into detail right now. So what I want to focus on and introduce you a little bit to is these two tools here. One is iPresto, that's the detection of biosynthetic subclusters in gene clusters to be able to predict monomers and precursors. And because I had the pleasure of following up Kiva's talk, I thought it would be nice to also say something about assembly line order and prediction of docking domain interactions, as the PKSPOP. So first, subclusters. What we see when we look at biosynthetic gene clusters, including NRPS and PKS gene clusters, is that there are often these operons or sets of auxiliary genes that are involved in biosynthesizing monomers. I think the Dean gave a number of examples in the glycopeptide gene clusters for the several non-proteinogenic amino acids that are being incorporated into these molecules. But very many PKS and NRPS gene clusters have these kinds of subclusters that are involved in making certain moieties that are incorporated into the molecule. And sometimes you also have different flavors of a subcluster, like in this case, you can have multiple related chemical moieties that are made by through the action of related subclusters, where you will, for example, have absence or presence of certain additional enzymes, like methyltransferase, for example. And we can also computationally identify these subclusters, and we've recently applied a text mining natural language processing algorithm called LDA, which is also used for topic modeling. So if you have newspaper articles, it would be able to find for topics like, okay, these words are related to football, these words are related to politics, etc. By looking at how words are co-occurring across different articles. And in this way, we can also find topics, biosynthetic topics of genes and domains that co-occur very frequently and more frequently than expected across gene clusters using the same natural language processing algorithm. And this LDA algorithm, and we also had a previously published statistical algorithm, and this way we can identify these subclusters that are co-evolving, that are apparently, these underlying genes apparently have a shared function. And in many cases, we can predict also links to a substructure if, for example, one of the gene clusters in which we find such a co-evolving subcluster has been experimentally characterized, and we find it back in the database. So Joris, who has actually worked on this, he was a master student in my group and will soon be joining us as a research associate. He actually also did a manual annotation of around 60 different subclusters across many different gene clusters, and he identified also the chemistry based on links to MIBig. So we'll have a pretty nice database of these subclusters, and we plan also on integrating this into ANTISMASH so that you can immediately see these links, but not only to the known ones, so to known subclusters, but also to subclusters which you do not know a function yet, so which would still enable you to see that there is a set of genes in your gene cluster that is probably involved in making some chemical moiety, and it's also found in certain other gene clusters. And in the end, if you use paired omics data, I yesterday introduced briefly for Michel Schorn, the paired omics data platform, where we have genomic data and metabolomic data of the same strains. You can also use this to find links and to find correlations. Like if we have a subcluster, for example, for this sugar moiety, do we find that co-occurring with motifs and patterns in tandem mass spectral data, which are also related to making a certain sugar, for example. And in this way, we hope that we're able to also link these patterns of co-occurrence in gene clusters and these motifs of genes in gene clusters with motifs from mass spectral data. So the final thing I want to briefly talk about is the PKSPOP algorithm, which is for docking domain interaction, because at some point when you predict the substrate specificity of NRPS and PKS modules, to be able to get an idea of the full structure, you need to know the order, of course, of the PKS and NRPS enzymes. And for this, you need to predict the interactions between the docking domains. And we make use of a lot of fantastic biochemical work by Kira and others to be able to see whether we could actually, at least for certain subtypes of these docking domains, the type 1A subtypes, to reliably predict the docking domain interactions. This is work by Jan Wang, a master student, work with myself and my colleague Aljan van Dijk, who's a specialist in machine learning, including applications on protein-protein interactions. And Aljan in his group, they had actually developed recently an algorithm, a general algorithm for protein-protein interaction prediction, which is an unsupervised algorithm, so it doesn't work with a predefined training set of known interactions, but rather it identifies through expectation minimization, it identifies co-evolution, so it sees which, in alignments, which sets of sequences, whether their diversity can be better explained by co-evolution compared to independent evolution. And then it gives a probability, how likely is it that indeed these sequences have been co-evolving instead of independently evolving. So we can make use of this by looking at applying this to all the possible pairs of docking domains and then seeing what are the probabilities of co-evolution for a given pair of docking domains based on their sequence alignments. And in the end, so Jan put this together into a more comprehensive algorithm that then uses this to predict the assembly line order. So what we first do is we take all the pairs of docking domains between the different PKSs of an assembly line, and we run this Ouroboros algorithm to identify evolution or co-evolution probabilities. So we get a matrix with probabilities. Then we can add a number of constraints. So for example, we see loading modules and we see thioesterates at the end, which means that we can add a number of zeros where we know that based on biosynthetic logic, certain interactions should not be possible. So this constrains the realm of possibilities a bit. And subsequently, after transformations, after probabilities to await, we apply the Hungarian algorithm to find the best set of docking domain pairs, which together have the lowest overall or the highest overall probability. So then we get, in this case, this range of combinations of docking domains. And then in the end, we get these pairs out here, and then we remove, for example, in this case, the pairs again based on biosynthetic logic, because this ends in a bioesterase. This starts with a new module, so this interaction we can remove. And then when we stitch these together into one assembly line order, we can predict, in many cases, a single protein order based on this co-evolutionary method. And we see when benchmarking this on known assembly lines, we see that for, let's say, roughly 80% of the cases, we can reliably predict a single assembly line order. So this is much better than random. So what is currently still there in ANTISMASH and what the previous approaches have largely been based on is by looking at specific pairs of charged residues, and actually in ANTISMASH currently it only looks at 2 pairs of residues. And what Jan found in her analysis is actually that the final model actually relies on considerably more than just these 2 pairs of residues. And I think this is in line with what Peter presented, that not only these pairs of charged residues matter, but there's a larger set of residues around it that indeed perhaps are involved in the stability or modulating those interactions. So in this way, by really involving the whole alignment of those docking domains, and then using this co-evolutionary model, we get a better prediction. And we really hope that we can extend this in the future to other types of PKS docking domains and perhaps also NRBSs. And then I think going forward into the future, what of course the dream is that we can put all these databases of G clusters together with biosynthetic information to be able to make new kinds of pathways, to do computer-aided design, to say, okay, we start, for example, with a certain G cluster that we get from a database, we find all related G clusters, and then we start swapping in domains, modules, etc. We're very excited that Mohammed, in collaboration with Helga's lab, is really starting to work on this and to make this dream a reality. So I'm really excited also about that collaboration. And with that, I'd like to close, and I'd like to thank my group and all the people who did all the hard work, also the funding agencies, as well as all the collaborators that have made this work possible. And yeah, thanks again to the organizers for this great meeting.
And some questions?
When you predict the docking domain matches, how often do the proteins not interact in a collinear fashion to the genetic arrangement? Is it usually the same as in the genome?
That's a good question. We have tested it on both collinear and non-collinear assembly lines, but I don't think we have actually quantified, like on unknown assembly lines, how often these would not be collinear. I guess if you just look at known pathways, a rough estimate, it would be like 2/3 of the times there is collinearity, or a bit more perhaps. I don't know, what would you think, Kia? Do you have an idea?
I don't, that was the same question I had.
I think there will be, because we had a set of non-collinear pathways, and there were a few dozens, but there is a few hundreds of collinear pathways, so it's a minority, but it's still a considerable minority.
Thanks for the great talk. I think if you compare all the development of ANTISMASH from the first to the new ANTISMASH method, and all the things that we can predict already, if we really look manually with all the different tools, right? How far do you think we really are from the full automation of predicting full structures, kind of correct structures?
Yes, that's a really good question, and I think there is still a long way to go, and we've actually on purpose being very cautious of predicting full structures with ANTISMASH, because we know there's so much uncertainty, and I think we could predict whole structures, but we think in many cases, in too many cases, they will be wrong. So what I think makes more sense for now, for the coming years, is that we become better at predicting structural features from gene clusters. So from a gene cluster, you would get a list of structural features and information on what we can reliably predict about, for example, their order. Let's say based on, for example, these docking domain interactions. But then I think predicting all the regioselectivity, etc., that is still very complicated. But I think if we can predict all the structural features of the substructures quite reliably in a natural product structure, then it should give sufficient information to really help you a lot with, for example, linking genes to molecules, linking the genomic data to metabolomic data.
But why do you think that is? Because if I look at gene clusters with full data at every enzyme manually, right? In a lot of cases, you can predict kind of, not in all, but in a lot, but much more than automatic. You can still predict structures better. So what's really missing to automate that?
Yeah, I think one of the things that is missing is, for example, better predictions of regioselectivity of tailoring enzymes. So if you have a methyltransferase, knowing where it's most likely to act. So I think a big step forward would already be if we can, based on, for example, chemical fingerprinting of these local interactions, and we can give probabilities to it. And we can say, okay, this methyltransferase is, let's say, 30% likely to do the methylation at this point in the structure, and maybe 20% at that location in the structure. But this will require, even being able to do this will require quite some technological advances, I would say.
This predicting co-evolution, you are now applying this on the docking domains. So it's a rather, let's say, that are small, short sequences. This is a very interesting approach also for larger interfaces. I'm just thinking about classical and alternative module boundaries. I mean, some of the interfaces I just described. Is it possible in terms of your computational power or what else you're thinking about, that you also try to apply this co-evolution approach on other interfaces, see between KSAP or ACPKS, so which interfaces co-evolve? As to my knowledge, this is classical and alternative orders. Now, in fact, we are talking here. There's still a bit of this systematic analysis missing. So could you?
Great point. Yes, so the question of Martin was, can you also apply the co-evolution method to other interfaces, for example, in PPS and RPS assembly lines? And actually, Jan did also apply the same algorithm to KS linker ACP interactions. So those, based on those alignments, but unfortunately, or maybe interestingly, she got very little signal out of these KS linker ACP interfaces. So the predictive power of the docking domains was much stronger than of the KS linker ACP interactions, which perhaps tells us something about, well, perhaps the docking domains play a bigger role in evolution in media.
Or, I mean, the interfaces are maybe not so clear.
That's also a possibility, absolutely.
My question might be a bit provocative, in the sense that what is the source when you say that you are using natural language processing from your PFAM import or something, or for your databases or the connections, let's say?
For the subclusters.
Exactly.
Yes, so we actually apply this, so we translate we actually use there the same approach as for big slice, so we translate a gene cluster into a string of domains and subdomains. And those strings of domains and subdomains are subjected to this topic model.
And are these somehow sifted manually also, or do you see that?
So the output, indeed, we curate manually to, for example, identify cases where we can link these topics to specific structural features.
Questions? Just a quick one. So how does your algorithm deal with, for example, unusual hybrids between pathways, or for example, split clusters where you don't have this nice grouping together of genes? Do you have any means to?
Yes, so that's a great question. So for example, if you get hybrid gene clusters, or you get gene clusters that are maybe merged together because they're right next to each other, I think the big skip algorithm probably is most elegant in handling this, because what you would get is, for example, a mixed or hybrid cluster would get links in the network with, for example, a family that only has one part and another family that has the other part. So they would be kind of hubs in the network connecting to other otherwise unrelated families. With big splice, because it's on overall clustering, it's a bit more difficult. But then you can make use of some of the new developments that we have, that Kyle-Lynn and Simon Shaw have entered into ANTISMASH-5, where each cluster is no longer only a cluster, we call it a region, and we actually have multiple underlying candidate clusters, which could be, well, sometimes you have two clusters that together are a region, but you could also do the clustering on the individual clusters that underlie a region, for example.
Just a naive question. As a person who's not a bioinformatician, if I wanted to use this software, do you actually have to collaborate with your group or other experts, or is that accessible?
No, we make all the software publicly available, and we also try to make it as user-friendly as possible. So if anyone has any questions on using it or you run into trouble, just shoot us an email and we'll be very glad to help you use the software. And yeah, for example, we also provide these web tools like now with BigFam, so you can actually interact with it through the web, which is also easier for many people than having to run programs or come online.
Your help desk 24 hours a day.
Not exactly, but we do our best.
Okay, with that, I think we can close the session, move to coffee. Thank Marnix again, and we'll thank my